Welcome everyone to this webinar about the launch of Chordoma Foundation Labs. My name is Josh Sommer. I'm the co-founder and executive director of the Chordoma Foundation, and actually, as of this month, a 16-year skull-based chordoma survivor. I'm really excited to be here with you today, along with my colleague, Dr. Dan Freed, who is our head of target discovery and translational research, as well as the director of CF Labs. So just a couple quick housekeeping notes before we get started. I just want to let everyone know that this webinar is being recorded and it will be available online if you want to run through it again, or if you have any additional questions after the fact. We do welcome questions throughout the conversation through the Q&A button. Dan and I will reserve time at the end for a Q&A session, so please do feel free to send in your questions. And then if you have any additional follow-up questions or feedback at the end, we'd love to hear from you at support@chordoma.org. So I think as many have probably heard through our communications online and on social media, the launch of Chordoma Foundation Labs is probably the most consequential step the Chordoma Foundation has ever taken to advance our mission. And we're really excited to share what's gone into setting up the lab, our motivation for taking this big step. We are going to spend some time today talking about the launch of the lab over the last 5 or 6 months, as well as give a view into the impact it is already having through some of the projects we've been able to take on and some of the projects that are in the queue, as well as talk about our vision for the impact the lab can have going forward. And finally, we'll end on Q&A. We have an hour allotted for this conversation. But Dan and I are available to continue on even after the hour if additional questions come in. So please don't feel rushed or the need to hold back your questions just because we're bumping up against the end of the hour. So I think first it's important to do a little bit of table setting and provide some context. Many of you probably know this well, but I think it's always worth coming back to what our guiding mission is. So the mission of the Chordoma Foundation has always been to improve the lives of those affected by chordoma and to lead the search for a cure. And we recognize that to carry out that mission, there are a number of things that are important. First, research to find better treatments, healthcare improvement to improve the way that care is delivered to patients, ensure that patients are getting the best care possible. And then patient services to help improve every step of the journey from diagnosis through survivorship. And together, each of these initiatives, each of these efforts is mutually reinforcing and ultimately allows us to move towards a vision and a future where patients are able to overcome the disease and maintain their quality of life. What we're going to be talking today is catalyzing chordoma research. And I think it probably goes without saying for those in the audience who are chordoma patients or loved ones of patients that current treatments are simply inadequate. It is definitely the case that surgery and radiation have advanced tremendously over the last several decades and are at this point incredibly sophisticated and in many cases quite effective. Many patients, if they receive the best care up front, can achieve many years of disease-free survival, and some patients are in fact cured. However, even with the best possible surgery and radiation, the majority of chordoma patients still eventually experience a recurrence. At the same time, surgery and radiation are oftentimes associated with significant quality of life impact, whether that's in the initial treatment setting or for recurrences. And I think the thing that looms large for many of us who have been diagnosed with chordoma is that if the tumor recurs, I think we all know that treatment options, unfortunately, are limited. Sometimes and oftentimes patients will have additional surgery and radiation, but at some point, those therapies become exhausted and no longer safe or feasible. Consequently, the median survival for chordoma patients remains at about 8 years. So what's needed? For those who are newly diagnosed, ideally there would be therapies that could do a better job even than current surgery and radiation at preventing recurrences. For those who do have recurrences or who have advanced disease, there's a real need for therapies that can eliminate, shrink, or durably control recurrent tumors. And throughout every stage of treatment, there are therapies that are needed that can improve quality of life. So thankfully, there are extraordinary advances happening throughout the realm of cancer care and oncology. There are big improvements in patient outcomes and quality of life happening in a number of other tumor types. And our job and what we are determined to do collectively as the foundation and as a part of this community is to make sure that chordoma patients benefit from all of these advances. There are opportunities to improve treatment and outcomes through better detection, through better local therapy, including surgery and radiation, as well as others, through better systemic therapy, as well as more precisely applying these various therapies in a personalized fashion so that patients get the right treatment at the right time. Among these various options, I think there is wide consensus among the clinical and research communities that the biggest need is for systemic therapies that can augment surgery and radiation and reduce the likelihood of recurrence. And importantly, once a tumor has gotten to the point where surgery and radiation are no longer good options or effective, can control tumor that has perhaps spread outside of the initial vicinity of the original primary tumor. Thankfully, there are quite a number of very powerful emerging systemic therapy options from a wide variety of new targeted therapies that are making accessible any number of targets, therapeutic targets that have previously been impossible to address with systemic therapies. There's been an absolute explosion in various immunotherapies for cancer, cell-based therapies, others as well, like radiopharmaceuticals that can precisely deliver radiation just to the individual cells, to the individual tumor cells, regardless of where they are throughout the body. And the reason that this is so important is that, as I mentioned, that when surgery and radiation, when local therapies are no longer safe and effective, there is a real dire need for these systemic therapies. And so that's what the Foundation's research focus has been on. It's not the exclusive thing, but it is the primary focus of the Chordoma Foundation's research strategy. The great thing is there are quite a number of different paths to potential systemic therapies. There are about 3,000 drugs that already exist, that are already on the market, and thousands more that are currently being developed. There's tens of billions of dollars that are being invested every year to develop new cancer drugs. And so the list of existing drugs, the toolbox, so to speak, is continually expanding. And so if we could find some of these existing drugs that could be repurposed for chordoma, that could provide, in fact, a near-term and quite rapid path to new therapies for chordoma patients. There's also the potential need to develop new drugs and quite a number of new technologies that are making that more feasible. So at the outset, we don't know which of these paths is ultimately going to lead to better treatments. But to answer that question, we have to be guided by the biology of the disease. So there's quite a lot of basic research that's needed to understand what drives the tumor, what are its vulnerabilities. And based upon that understanding, it can lead us either to rapid or quick repurposing opportunities, or can provide rationale and indicate a need to develop new drugs. But ultimately, all of this converges in the need to test therapies in clinical trials. And I think that it's important to recognize that because chordoma is a rare disease, there's some unique constraints that are imposed on running clinical trials for chordoma. There's only so many patients that are out there, and so that limits the number of clinical trials that can ever be done. Obviously, resources are constrained, and so for a rare disease like chordoma, the bar has to be really quite high to justify investment in clinical trials. Additionally, it's very likely that there are going to need to be multiple shots on goal. Tumors are complex. It is almost universally the case that to really control, if not cure, cancer requires a combination of therapies. Sometimes that is concurrent, and sometimes that is in sequence. Tumors are continually evolving, and so it may be the case that tumors develop resistance to one therapy, and you need to be able to have a backup and a backup and a backup. Moreover, there's heterogeneity between patients. Not all chordomas are the same. And even within tumors, there's heterogeneity. And so it is very likely that we're going to need to not run this path once, but run this path multiple times. And then the reality is that some of these efforts are not going to pan out in the clinic. As smart as we think we are, and as good as our ability is to predict, at the end of the day, across the field of oncology, the success rate of drugs in clinical trials is not tremendously high. And so we must anticipate the need to, as I said, to run this path from discovering some new aspect of the biology of the disease, and that leading to some hint of a potential therapeutic concept, and run that down all the way to the point of a clinical trial. Now, I think that it would be fair to ask, is this feasible? When the foundation started, Chordoma research was at an absolute standstill. There were just a handful of researchers working on studying Chordoma. There were no new therapies in development. I think we all know that the challenges for research for rare cancers are multiplied. There's not a large market incentive to drive investment on the part of pharmaceutical companies. Because of the rarity of the disease, there are oftentimes not that many researchers who are interested. There can be quite a number of logistical challenges. And even if all that was solved, even for the most common and well-resourced cancers, it is unfortunately the case that research oftentimes does not move with a sense of urgency that matches the urgent need of patients. So our challenge from day one, recognizing all of those things, I think we have not been naive to those challenges, and we've not been naive to the fact that this is going to be an uphill battle. And in fact, that achieving the treatments that we need and desire is certainly not the norm for rare cancers. But recognizing that, is there a way? Can we find a way to engineer a research system that can drive progress fast enough for current patients to benefit? We need treatments in a matter of years, not decades. Those of us who are facing this disease now are in a race. We have an urgent need. And while we certainly hope that patients in the future will benefit, at the end of the day, this is very personal, and we need to find a way for patients who are dealing with this disease today to benefit from investments of research. So, when the foundation was starting, and certainly at every step along the way, we have constantly been learning and trying to understand, what does it take to make research go? And what we have learned over the years is that there is a whole array of inputs, a full complement of inputs that are required and that are necessary but not sufficient to advance new therapies. So I think it's probably obvious that you need a certain set of expertise. You need a certain set of research capabilities. You need scientific resources, things like tumor tissue, cell lines, animal models, to be able to actually study the disease, understand its biology, and translate those insights into a strong concept that can be tested in the clinic. You need patient participation to contribute data, to contribute biospecimens, to participate in clinical trials. Of course, you need funding to make this all go. And at the end of the day, you also need initiative. You need someone or an entity or entities to ensure that things don't slip through the cracks, that important questions and needs and opportunities get chased down, and that research continues moving forward proactively rather than waiting for and hoping for someone to get around to it. If we look around at what is the norm for disease-focused foundations, patient organizations, certainly when the foundation was starting, I think the obvious thing to do is to raise money and fund research, and that is definitely a tried and true model. However, I think what became very apparent is that funding is necessary but not sufficient, and no amount of funding invested into a system that is not configured for speed and progress, and that is not does not have the full complement of additional inputs that are required, no amount of funding is going to make research go. And so based upon that recognition, over the last 15 years, we have tooled and designed the foundation and through the foundation, marshaled this full complement of resources in concert to really step on the gas of Chordoma research. We've over the years recruited or helped to recruit over 300 researchers in academia and industry. We've developed tumor models, cell lines and mouse models, and made them available through centralized repositories. We've invested about $15 million in research and leveraged the best of our ability to estimate about $25 million in additional funding. We have built a strong and vibrant Chordoma patient community and not only connected with the patient community, but also built a whole suite of services to provide education, navigation, and support. And so all of those things together have contributed to a huge uptick in the pace of research progress. But what we've observed is that even with all of those things in place, while research is certainly moving, there are 7 therapies that have made it into clinical trials in the last 15 years as a direct result of the research that the foundation has supported. Even so, the pace of progress is still not fast enough. And what we're observing is that, and that is in large part due to structural limits imposed by the way that biomedical research is organized. And I have to say that this was not at all evident or obvious at the outset, but has really crystallized and become clear over the years. And so I want to walk through exactly what this insight is and what we've learned as to what are those structural barriers that limit the pace of research progress. Because understanding them, I think, is really critical to being able to overcome and develop solutions to get past them. So to advance any one of those therapeutic concepts that I alluded to previously, whether it be a repurposed therapy, whether it be a new drug, there's a whole range of expertise and capabilities that are required, from discovering and validating new targets to really understanding the biology of the particular signaling pathway or protein that's implicated, to testing drugs in cells and animals and understanding really how that drug works. So in all cases, there is a kind of an understanding, an expertise, and a set of capabilities specific to chordoma that are needed. But additionally, there's expertise and specialization that's needed in target biology, in various sophisticated technologies, and in specific drugs or therapeutic classes. Now, because of the way that academic and industry research is typically organized, this expertise is oftentimes siloed. Researchers oftentimes specialize in one or maybe two of these particular areas, but rarely, if ever, are these sets of expertise and capabilities housed within a single lab or even within a single institution. And so that necessitates, even for an individual project, the involvement and initiative of many investigators. And then that's only multiplied because there is a need to pursue multiple therapeutic concepts in parallel, and as I said, take multiple shots on goal. And so what this means is that there is a need for many researchers to take on chordoma. We've been tremendously fortunate and we are tremendously grateful to those 300 plus researchers who have gone through the process of becoming involved in chordoma research. But the reality is that very few labs start with experience studying chordoma. And what that means is that to go from a standing start, to go from having never studied chordoma before, there's a huge learning curve. There's a lot of startup costs, and there's a lot of time that it takes to get projects going. Moreover, once labs do have established a chordoma research project or capabilities, because of the barriers and challenges and obstacles to collaborating between various institutions and between companies, oftentimes those same capabilities are duplicated every time an additional researcher becomes interested or decides to take on a chordoma project. And the reality is that as science advances, as we're learning more about the biology of the disease, there's a continual need for new expertise at the bleeding edge of chordoma biology and therapeutic development. And every time a new researcher comes into the field, what we're seeing is that capabilities have to be duplicated. Moreover, most researchers or teams are specialized, and they focus on a single or maybe a few different therapeutic concepts. And what that means is that experiments are done in small batches, they're done infrequently, and there's no opportunity for economies of scale ultimately leading to low experimental efficiency. And sometimes, even when there's interest, researchers lack the facilities, the know-how, or the focus to pursue worthwhile concepts, ultimately leading promising concepts to be completely stranded. Some examples of this are, it is a kind of a common theme we've seen where medical oncologists who treat a lot of chordoma patients, really understand this disease, understand its biology, will have really good ideas, but don't necessarily have a lab of their own with which they can pursue those ideas. And so at a certain point, what we've realized is to move faster, we need to build a faster engine. No amount of funding, no amount of willpower, no amount of patient participation in this current system will make it go faster. We must build a faster way to get research done. And so what we observed, and this goes back 7 or 8 years now, is that there are a common set of core chordoma-related research capabilities that many labs, if not most labs, need to establish to advance therapeutic concepts for chordoma. And so the question that we asked ourselves is, why do we have to continually build those in individual labs and duplicate that effort, do it at small scale, low experimental efficiency? What if we created a shared centralized platform, research platform, that could take advantage of the economies of scale to increase the speed, efficiency, and feasibility of key research? And so the idea here is the Chordoma Foundation could build these capabilities and build a set of chordoma-focused expertise, and then work with partners, whether that be in academia or industry, who have specialized expertise in biology, in technology, or particular therapeutics, and allow those researchers to quickly and easily and more efficiently pursue their bright ideas and their projects in the context of this centralized research platform. And so our first foray into creating a centralized research platform was in 2015 when we set up the Chordoma Foundation's drug screening program. At the time, what we noticed was that there were a lot of really promising concepts that were bubbling up in labs across the world. And they were not moving into clinical trials because what was missing was being able to demonstrate that those particular therapies were actually capable of shrinking or slowing the growth of tumors in mice, which is oftentimes a critical data point that's needed to be able to justify a clinical trial, to be able to get companies to either provide funding and at the very least to be able to provide drug for that trial. And so the idea was, could we centralize these experiments, which are time-consuming and costly to do at a small scale in individual labs, and make this service available to the research community? So indeed, that's what we did. One step at a time. Started off with a couple different models, and ultimately over the years have assembled about a dozen chordoma mouse models in which we're now able to test promising concepts. And what we're seeing is that indeed, as hoped, rather than taking oftentimes a year or more to get experiments going in in a typical academic or industry lab, because the experiments are basically already up and running, the models are already warm, we can go from concepts to experiment in weeks or in a couple of months. The experiments take the same amount of time, and sometimes we are bound by confidentiality and must respect the needs of researchers or companies to embargo sharing the results. But other times we're actually free to share the data immediately so that other labs can act on it promptly. And what we've seen is that this has just absolutely catalyzed a tremendous increase in the speed and throughput of drug screening experiments. So to put this in context, when this program started, there had been just a handful of therapies that had ever been tested in mice before. And over the last 7 years, we've received 111 concept nominations from researchers and companies across the globe. 86 of them have passed our review process. We've begun testing 79 of them, nearly 60 are complete. And of these, 6 have now advanced to the point of clinical trials. 5 of those clinical trials have actually gotten started. So tremendous catalytic impact of the drug screening program. But what we're observing now, what we've seen over the years, is that there is a wide variety of additional capabilities where we're seeing a similar bottlenecks and a similar need for greater efficiency and speed. And so this really spans the entire preclinical R&D continuum, everything from characterizing and validating disease models, validating new targets, work to develop new therapies targeting Brachiary, which is the Achilles heel of chordoma, as well as comparing drugs, for example, head-to-head in cells as a way of prioritizing and informing the design of those experiments in mouse models that I described. And we'll talk a little bit more, we'll give some examples of these when I turn it over to Dan here. So, we've been noodling over the years as to how best to address that need. Certainly there's a variety of ways that it could be done. It could, could involve partnerships with designated academic labs. It could involve core facilities at academic institutions. It could involve working with contract research organizations. And we've very, I think, intentionally and systematically evaluated all of those options. And in doing so, we were inspired and informed by a couple of different precedents, both with the Cystic Fibrosis Foundation and the Therapeutic and the ALS Therapeutic Development Institute, which are peer organizations, granted larger and older peer organizations, but nonetheless addressing a similar need. These organizations set up their own labs to provide their respective research communities a variety of centralized capabilities, as well as to be able to drive research of their own. And the impact has been extraordinarily catalytic. And so we said, what if we could apply that same model to Chordoma, recognizing that having our own lab would provide the greatest nimbleness. It would allow us to build in-house expertise that wasn't necessarily possible through contract research organizations. And it would be certainly more cost-effective than working with contract research labs. So after a great deal of planning and a lot of advice and input from a great number of advisors, we took the leap earlier this year and launched CF Labs. This is the first lab 100% dedicated to chordoma research. And as far as we know, it's actually the first of its kind for any cancer. So there is certainly an element of trailblazing here and a lot of learning. The premise here is to specialize in a variety of chordoma-specific experiments in chordoma cells that Dan will talk about momentarily. And by doing those multiple times to create economies of scale, drive down the cost, increase the efficiency, and increase the robustness. We're very fortunate, first of all, to have Dan on the team. And secondly, to be located in a city where we have access to biotech incubator space that provides essentially a turnkey opportunity to stand up a lab with very minimal initial capital investment. And so starting in January of this year, we occupied a bench in biolabs, which I'm going to turn it over now to Dan to talk about. And I'll just take a moment to give Dan a little bit of an intro. So Dan joined us about 1.5 years ago as our head of target discovery and translational research, with just an incredibly ideal background spanning academia, a large biotech, a startup biotech, and has actually had experience starting multiple different labs. His expertise has spanned all the way from basic research through translational research. And so we feel just extraordinarily fortunate to have Dan on the team and to be focused day in and day out on understanding the biology of the disease and then rapidly translating that understanding into new therapeutic concepts. So Dan, I'll turn it over to you, and Dan will talk about the launch of the lab and where we're heading from here.
Great, thanks, Josh, for the kind introduction. And let's see if I can share my screen here. Okay, well, it's great to be here, and I'm excited to tell you all about CF Labs, which we launched in the first quarter of this year. And so in the middle of January, we hired our first scientist. That's Nindo Punturi there, who also has just an incredible background that includes the launch of new labs. By the end of January, we had imported 21 Chordoma cell lines and put the first 5 of those in culture. And by early February, we had the lab fully stocked and set up. And here's a picture of me and Nindo standing next to a lab bench that is just fully equipped and ready to go. And at that point, we began establishing various experimental capabilities. And that put us in position by the beginning of March to collect our first data and obtain our first results. So that's me and Nindo standing at the computer screen discussing some of our first results. And importantly, we also implemented robust systems that will allow us to scale CF labs as we move forward. So in terms of experimental capabilities, so far we have established, and importantly, we've optimized a number of these that include cell cultures, drug screening assays, assays to understand how drugs work or don't work, as well as cell imaging. And one important thing that CF Labs is going to do is generate optimized protocols. And we're going to share these protocols with the Chordoma research community to try to standardize these assays to the extent possible. Just to give you a little bit more flavor about what that means. So one of the important experiments that we've established and optimized so far, and we're still indeed in the process of doing, is drug assays. And in order to kind of accurately evaluate and compare drug activities from experiment to experiment, it's important to optimize several parameters, most notably the number of cells that are used in an assay and then how long that assay extends for. Now, this technical information has never been systematically reported for Chordoma cell lines. And so what you have in the literature are a number of publications where it's kind of difficult to know whether there's an apples to apples comparison for results that are from drug assays. And so what we're doing is we're optimizing this information for every Chordoma cell line. And we're going to share this information in order to promote standardization. And below here is just a little snapshot of one of those experiments that we've been doing. And what we found was that the blue line here, 3,000 cells, kind of gave us the number of cells that is sort of the ideal target range for starting an assay. And here we found that we didn't do the assay for a long enough period of time. And that's why it's important to do these optimization experiments. But by the shape of this curve, we know that we need to extend this assay duration for another day and a half or 2 days. And so we're in the process of verifying that. And we're going to do this for every Chordoma cell line so that we make sure that we can kind of establish these parameters and standardize things moving forward. And so by doing all of this, by establishing these capabilities, these core capabilities, and optimizing them, and then promoting standardization, among other things, CF Labs is able to offer kind of a much faster and cost-efficient and throughput-efficient alternative for conducting core experiments compared to what would otherwise be possible if we were to outsource these experiments to our academic or industry research partners. And so in that way, we feel that we kind of complement the strengths of our academic and industry partners. So we can kind of take on these kind of core preclinical research capabilities that are Chordoma specific and allow our partners to focus on doing what they do best. And that's focusing on specialized biological, technological, and therapeutic expertise that they've developed in their respective labs. And so we can then be opportunistic in our collaboration with outside experts rather than exclusively relying on them for everything. So we imagine kind of 3 different roles that we can play in our engagement with external collaborators. So at CF Labs, we can perform projects that enable collaborators to do experiments that they otherwise wouldn't be able to do efficiently or quickly or easily or possibly even at all. So we can play a supportive role. We can also play a role in inducing further work by researchers or companies. So we can generate preliminary data that can justify and de-risk further work by other labs that might not prioritize chordoma or a certain therapeutic concept for one reason or another. And third, we can take the bull by its horns and be disruptive and drive concepts forward. So we can pursue internally conceived projects or those that have been neglected or stranded by the research community, and kind of take a no therapeutic concept left behind approach to development in chordoma. So this is just a snapshot of the 5 projects that we currently have underway at CF Labs and the various roles that each of these projects kind of align with. And I'll just provide a couple of quick examples of a support project, an induced project, and a project where we're driving a concept forward ourselves. Just to give you a sense for some of the things that we've been doing in the early days of CF Labs. So for a support project, we've been working with a pharmaceutical partner to study their PI3K mTOR, their dual PI3K mTOR inhibitor in our drug screening program over the past year or so. And the data has looked incredibly good. And so we brought this therapeutic concept to our medical advisory board earlier this year. And they were very intrigued by the data that we had generated, but recommended that we do some further due diligence in order to compare this drug to its clinical stage competitors, and as well as to a similar drug that's previously been tested in a chordoma clinical trial, just to make sure that we can kind of put our best foot forward and be confident when we go into a clinical trial that we're using the best-in-class drug as it pertains to chordoma. So this would have otherwise taken us months to do before CF Labs, but we could hit the ground running, and we performed this additional testing and generated results within 6 weeks. And what we concluded was that the red drug here, drug A, appears to work the best. And I'm keeping these drug identities confidential for now because of the nature of our engagement with this pharmaceutical company. But suffice it to say that we were able to identify the best-in-class drug so that we know that we can move on to the next step and be confident that we're taking our best shot on goal. So in an inducement role, we have a project that's ongoing with a view to exploiting a new form of cell death. 5 years ago, in 2017, we funded researchers who identified 2 genes that are essential for the survival of chordoma. And what was interesting about these genes is that they were associated with a vulnerability to a certain form of cell death. But this was not really further pursued due to a number of reasons, including strained bandwidth and competing priorities. And so 5 years went by and CF Labs was launched, and we began testing this interesting hypothesis within weeks. And this is sort of an induced role because this data will support initial or ongoing conversations with 2 different biotech companies to try to further explore this new therapeutic strategy in chordoma, provided that the preliminary data looks promising. So we did the experiments and the data did look good. We were able to target this pathway using what's called a preclinical tool compound. So it's not a drug in and of itself, but it can be used to try to identify whether a therapeutic concept looks promising. And we saw that we could induce this form of cell death in various chordoma cell lines, and that we could reverse this effect to confirm its mechanism. So this allowed us to confirm that this appears to be, in fact, a vulnerability in chordoma. And this will support our initial or ongoing conversations with pharmaceutical partners. And finally, we have a project ongoing where we're driving a therapeutic concept forward. And this therapeutic concept that I'm going to discuss has to do with EGFR inhibition. So EGFR has been a validated therapeutic target, chordoma, for quite some time now. And afatinib and cetuximab, 2 EGFR inhibitors, are now in phase 2 chordoma clinical trials. So the preliminary data and preclinical support behind this concept is incredibly strong, but we still don't know which patients are most likely to respond to treatment And for those who do benefit from EGFR inhibition, why therapy might eventually fail. And so the impact of understanding the answer to this question is to allow us to guide personalized care by sparing likely non-responders the side effects of an ineffective treatment, or to inform combination or sequential therapy approaches for folks who do benefit on EGFR inhibitors, but then eventually go on to relapse on treatment. And so we've been taking every chordoma cell line that we have in culture in the lab and treating them with EGFR inhibitors such as afatinib to try to understand how they respond to afatinib and why they're responding in that way. And well, it's still early right now, but the preliminary data are suggesting that 3 classes of chordoma cells appear to be emerging in terms of their response to afatinib. Those like JHC7 that are resistant to afatinib. So shown on the top left here are untreated cells, and then on the bottom are cells that are treated with afatinib, and the cells continue to proliferate, albeit a little bit slower. Cells that are sensitive to afatinib and die. That would be UCH1 cells, where on the top, the untreated cells continue to grow, but the treated cells just shrivel up and die. And then there are other cells that appear to be highly sensitive to EGFR inhibitors, but they just freeze in time. They literally stop growing as soon as you add afatinib. And one example of that type of cell is UCH CF365, shown here on the right. So that was a really interesting emerging observation for us, and it prompts 3 important questions that we can continue to pursue in CF labs. For example, why are some chordoma cell lines sensitive to afatinib while others are resistant? Why do some sensitive chordoma cells die, whereas others just stop proliferating? And if we can figure out the answer to that question, we might be able to develop combination therapy approaches that can induce cell death, and that might turn somebody that would respond with stable disease into somebody that would respond with a partial or complete response clinically to afatinib. And third, what are the mechanisms of acquired drug resistance in sensitive cells? And we're already getting a little bit of a window into this question. We have seen that in the case of UCH1 cells, whereas many cells die in response to afatinib treatment after a couple of days, those cells are exemplified by the red arrows in the figure to the right. Other cells undergo a profound morphological change. And this type of morphology change is associated with a specific resistance mechanism to EGFR inhibitors in lung cancer. And so we might be able to borrow all of the knowledge that's accumulated in lung cancer with EGFR inhibition in lung cancer and kind of repurpose that knowledge for chordoma to understand whether similar things are going on here and whether that might point to a potential combination therapy approach in chordoma. So these 3 important questions represent promising new avenues of investigative pursuit, which we are going to chase down in CF labs in the near future. And we're going to combine this emerging data with results from our model sequencing projects to develop predictive models to try to understand which patients are most likely to respond or not respond, which patients might be candidates for combination therapy or sequential therapy. And we can validate these predictive models with existing drug screening program data and with possibly patient samples from the ongoing afatinib and cetuximab clinical trials. So as we generate this data in CF labs, one thing that we really want to emphasize is data sharing in order to promote and catalyze further research and further progress in the chordoma field. One thing that we wanted to define was the optimal balance between rapid data sharing and ensuring scientific rigor. So we don't want to share data before we're confident that the result is robust and reproducible. And so we've established guidelines that experiments must be repeated to ensure the reproducibility of the results. The data must lead to a well-defined conclusion, though it can be narrow in scope. And the conclusion should ideally be self-consistent, such that the same result can be reached through at least 2 different experimental approaches. So once data for a specific concept reaches these thresholds, we'll share that data with the rest of the Chordoma community. And optimized protocols can be shared immediately with the scientific community, though, as soon as they're kind of developed and optimized. Okay, so so we've built these capabilities. We've launched CF Labs, and just to kind of riff off of the building a faster engine analogy that that Josh spoke to earlier, we're seeing that the demand that we have can't possibly be met with our current capacity in CF Labs. So we just have a ton of demand that's starting to build up. And so what we're trying to do now is shifting our focus to pushing our engine a little bit further and to kind of going into the scale phase. And so what does that look like? Well, our envisioned development tiers are our first launch phase. So that's where we are right now. We have myself along with one bench scientist and one lab bench at Biolabs. And we're undertaking our first 5 projects along with optimizing and establishing a variety of foundational experimental capabilities. We want to move into the scale phase as soon as possible, where we can add a second bench scientist. That will allow us to undertake additional capabilities, such as supporting brachiary drug discovery. And in this phase, we'll be able to decrease the project queuing time, increase our efficiency and throughput, and theoretically undertake more than 10 projects a year. And once we kind of move through the scale phase, we'll eventually move into kind of the deepening or expansion phase, where we aim to add PhD level scientists in each functional area to lead each key area of research, such as brachiary or immunotherapy or drug repurposing. We'll be in a position at that point to apply for NIH funding. And that's really when the full potential of CF Labs will be realized. So really appreciate your attention. And I think at this point, Josh and I would be happy to take any questions.
Wonderful. Thanks, Dan. So we actually have some really good questions coming in. I'm going to take them roughly from the top here. So the first question was about what's the current state of brachiary drug discovery? And I think, how does CF Labs fit into that? I think the short answer on the current state of brachiary drug discovery is that there is a lot of progress being made towards the development of a set of research tools. So Dan described earlier this concept of a tool compound, which is kind of chemicals that are precursors to drugs that can be used to kind of validate a particular target, whether that be in this case, chordoma or other tumor types, but importantly, can also serve as the starting point for drug discovery. So right now, that work is being done through academic partners. I think it's important to mention that we don't have currently, nor do we in the near term envision having chemistry capabilities in the lab. But to advance a drug, there is both a need for chemistry as well as biology capabilities. And this is something that we've thought a lot about, and we envision CF Labs playing an important role in the subsequent stages of brachiary drug discovery. And in fact, stages that are coming up somewhat quickly. So Dan, you've given this a lot of thought. Do you want to add to that or embellish in any way?
No, I think that's right, Josh.
Okay. Next question is, how can patients help make CF Labs a success? Well, this is I think a question that we really appreciate. So our hope and our vision with CF Labs is to get to a point where research is no longer constrained by the limits of the way that research is structured in academia and industry, but rather to be limited only by the biology of the disease. So how fast do the tumor cells grow, as well as the resources that we have at our disposal. And so we're really, really fortunate that over the years, we've acquired the cell lines that are needed to make all this research possible. And That is thanks to the tumor donations of many patients. So that's very important. Going forward, basically the structure has been built. We've demonstrated over the last 5 months that this is feasible, and we're seeing demand. And so now really the missing ingredient is resources. So with additional resources, we can take that next step of going from launch phase to scale phase, and hopefully do that as quickly as possible. And delighted to talk more about that if there are any additional specific questions on what level of resources are needed and kind of what that plan looks like. So the next question, Dan, I think we're seeing these in the same order. It's around partnering with drug companies. Would you like to take that one?
Oh, sorry, I don't think we're seeing these in the same order.
Okay, so I can read it. So it's when you partner with pharma companies, how do you handle ownership of IP that's generated jointly with the pharma partner?
Gotcha. Well, that's kind of handled on a case-by-case basis, and it's usually dictated by the contracts that are set up with each specific pharmaceutical partner. So, typically existing IP, of course, Chordoma Foundation makes no claim to any existing IP. If new IP is developed jointly in the during experimental work in CF labs, for example, or in the drug screening program, then I think there are scenarios where we can be co-inventors, but the finer details are usually spelled out in the testing contracts. Josh, do you want to add to that?
Yeah, I think it's just some important context is that the opportunities to create IP are extremely limited in the context of repurposing existing drugs, which is basically everything that's happening to date. So those drugs are basically already covered by a kind of wide array of patents, both composition of matter as well as method of use patents. And those method of use patents would be so broad as to already include Chordoma. So the goal here is not to be generating IP. The goal is to be moving as quickly as possible. And in fact, that is a difference between Chordoma Foundation and a lot of academic institutions. Oftentimes, it can be quite difficult for companies to collaborate with academic institutions because academic institutions are oftentimes very aggressive about wanting to kind of hold on to any IP or make sure that they can hold on to any IP, even if the likelihood of that IP being generated is exceedingly low. And so there will be instances in particular around Brachiary drug discovery, where there may indeed be IP generated, but at this point, it's exceedingly rare, if not, it has not happened actually to date with our drug screening program. Okay, so the next question is, will pediatric chordoma research be helped by CF labs? And the answer is a resounding yes. And that is thanks to investments that have been made over the last several years to develop pediatric chordoma models, both PDX models as well as cell lines. Dan can speak to this. In fact, some of the data that Dan showed, I believe, was actually in pediatric models.
Yeah, that's right. In fact, one of the first 5 projects that we have ongoing in CF Labs right now is not exclusively, but primarily focused on pediatric cancers. So it's focused on poorly differentiated chordoma primarily, which is enriched in the pediatric population. So absolutely, yeah. And
so the idea is for each of the concepts that are tested, they need to be tested in a number of different models. So you want to make sure that the results are generalizable, that they aren't specific or idiosyncratic to one model of one person's tumor. And so sometimes there are very specific reasons why certain models are chosen for certain experiments based upon the particular characteristics of those models. But just as a general proposition, our aim is whenever possible to include pediatric models alongside other models. And not, frankly, not just pediatric models, but to include a breadth of different models representing the diversity of the disease, including skull base and models of other origins, recurrent and metastatic, et cetera. So yes, very much a engine of discovery for all subsets of the chordoma patient population. Next question. This is the first lab of its kind for any form of cancer. Do we expect other cancer foundations to follow in our footsteps? I think that we are very much in learning phase here, and this, hopefully can serve as a case study and an exemplar for what's possible. I think the story is not yet fully written. So, once we're able to scale and once we're able to achieve the full potential of CF labs, I think, we will at that point know whether the impact is what, we have thought and hoped that it would be. But certainly at the outset, I mean, just as Dan mentioned, he gave the example of the question we were able to start answering, get a preliminary answer to within 6 weeks. I mean, we're already seeing just a dramatic shortening of the timeline to getting answers to important questions. And we know that the same scenario plays out in, many, if not all, other cancer types. So one would imagine that this model would certainly be transferable as well to those tumor types. The next question is, how will CF labs help patients in the short term? I think the overarching answer is by shortening the path to bringing new therapies into the clinic. There are quite a number of promising therapeutic concepts that have emerged from labs all over the world. And there's a certain set of steps that you have to go through to translate that concept into a clinical trial, which ultimately is, where patients benefit. And I think as we've seen over the years, that sequence of steps oftentimes is slow, inefficient, sometimes completely infeasible. CF Labs basically breaks down the barriers and makes that process efficient, seamless, faster, and such that our speed is only limited by biology and resources and not by the kind of overarching structure of academic and industry research.
Yeah, and just to add to that, I mean, I think it's our ability to be nimble and agile. And, one of the examples I provided in the presentation of, an experiment that would have otherwise taken months that took us 6 weeks to do, to ensure that when we move into the clinic and do a clinical trial, we're going to be doing that clinical trial with the best in class drug for that specific therapeutic concept. And thus the clinical trial has, the best chance to succeed. So I think, a lot of these kind of core foundational capabilities that we're establishing in CF labs that we would otherwise have to outsource to researchers are going to allow us to grease the skids and be a lot quicker and more efficient, but also smarter in how we translate concepts from bench to bedside. So there's tremendous potential for helping patients in the short term as well as in the long term, I think.
Yeah, absolutely. Okay, so there's a question about data sharing, and there's a balance between respecting the needs of confidentiality of our biotech and pharma partners while also trying to get data out into the public domain so that other investigators and ultimately patients can benefit. So Dan, you want to speak to that at all?
Yeah, that's a great question. I think so. One of the things that we do is I think really what this question speaks to is validating a new therapeutic target or a therapeutic concept. And so when there are commercially available drugs for a given therapeutic strategy, we can purchase that and perform the target validation experiments ourselves. And then we can widely share that with other researchers and academics and drug companies alike. And they can make their own decision, having that data in hand, whether it's worth pursuing with their proprietary technology or drug. But in general, we, like Josh said, we feel like there are, it's good to take multiple paths. And so we don't exclusively collaborate with industry partners necessarily. We want to take every path forward to evaluating a concept when there's mutual interest to doing so.
Yeah, I think that's spot on. Okay, there's two related questions here about funding needs and funding plans. So, back of the envelope, where we are now, the kind of launch phase at this level of scale, we're talking all in about $250,000 to $300,000 per year to operate at this phase. Now that included some initial startup costs. So if we were to continue at the same scale in subsequent years, that would be probably about $200,000 per year. Where we want to get in the kind of scale up phase, so that now you're talking about adding an additional scientist, an additional bench. Basically, that's about $400,000 to $450,000 per year to be able to operate, to be able to take on all of the experiments that we foresee coming down the pike. And then, to expand and hire PhD level scientists, that could be anywhere from, it goes up from there, but kind of where we envision $500,000 to $800,000 per year. Now, to get to that point, we certainly envision needing funding from the NIH, potentially having sponsored research from industry, but to continue at the current scale, and ultimately what we want to do is move forward into the scale phase as quickly as possible. The need really is for individual and philanthropic support. And so I think what we're seeing is that there's really no higher leverage investment that could be made to advance chordoma research. And there's really no greater way that those of us who are impacted by this disease can influence the outcome for ourselves or those that we care about. So we think that there is certainly a compelling case and certainly a great need to invest. And we're very grateful to the donors who have provided the seed funding to get this started. I think it really took vision, and there was some risk taking involved. But we're seeing, thanks to Dan and Nindo's capabilities and experience, that we've been able to stand this up extremely fast. They have built a very robust and scalable foundation, and so now really the rate limiting factor is resources. All right, so we're at the hour, but there's quite a number of questions coming in, which is awesome. So we're going to keep going. So there's a question: do you need tissue from a patient in order to determine what will or will not work for them? Dan, you want to take that?
Yeah. So this is more of a clinical question, something that wouldn't necessarily be answered in the lab, a preclinical lab like CF Labs right now. But the answer is yes. You need a tissue sample in order to determine what will or not work on them. What we're trying to understand in CF Labs is, how do you understand what will or will not work on a given patient? And so right now we have disease models such as mouse models and what are called chordoma cell lines that are cells derived from a patient's chordoma tumor and kind of adapted to grow on petri dishes in the lab. So we can use these sort of what are called disease models to try to understand for a given therapeutic concept what are the factors that allow us to predict what will or will not work on a patient. Once we understand those factors, the hope is that doctors, oncologists can use tests in the clinic, and a patient could have a tissue sample taken, that test could be run, and in real time, you could basically, it could guide personalized care. I hope that answer makes sense. Josh, do you have anything to add?
Yeah, I would just add that there's a complementary set of projects that we're supporting that are not happening in the lab. So I think it's important to recognize that the lab is not the be all end all. This does not obviate or replace all of the work that must be done in academic settings, but rather makes a lot of it more efficient. So in particular, there's 4 different projects that we're involved in or supporting right now to try to generate a fuller picture of the biology of the disease using large and very diverse cohorts of chordoma tumor samples. And the idea there is to be able to subtype chordoma at a molecular level, and then ensure that the models that we have are representative of the full spectrum of molecular subtypes. And so in that sense, once that is, as that knowledge accumulates and as we become more certain that the models are representative of all the different relevant subtypes, then you could start to match therapy to subtype in the models and then determine, in the clinic what molecular subtype a patient falls into. I think it's also worth noting that some therapies are going to be ubiquitous or generalizable across all subtypes. So we think that Brachiary drugs, for example, are highly likely to work across the majority, if not all, of the entire chordoma patient population. Whereas some of the concepts that Dan is working on now and that were shown in the slides, they're intended to address 10 or 15 or 20% of the patient population at a time. And then it'll be critical to take a tissue sample and determine whether a given patient falls into that 10 or 15 or 20% category. All right, question here about cetuximab. Is the response related to the genome structure of the tumor? Dan, do you want to address the mechanism of response to cetuximab?
Yeah, the answer is we don't fully know yet, but that's likely to be a key component of determining the response. Cetuximab is approved in colorectal cancer, and we know that EGFR expression levels are important for determining response to cetuximab. We also know that the genome structure, again, in colorectal cancer, is important for understanding which patients are not likely to respond to cetuximab. So I wouldn't be surprised if a similar story ended up playing out in chordoma. It's something that we're very interested in studying as part of the EGFR inhibitor project in the lab right now. And I wouldn't be surprised if there's a component of the genome structure that is going to be important in determining who responds and who doesn't. So great question.
Yep. So the next question is, NIH extramural funding is awarded to researchers at universities, medical schools, and other research institutions. Where does CF Labs fit within this organizational landscape? Well, it's definitely true that the majority of NIH funding goes to universities and medical centers, but pretty much all of the calls for funding also include other research entities. Sometimes it's limited to nonprofit research entities. There are standalone research institutes, RTI, SRI, Van Andel come to mind, any number that are independent nonprofit research institutes that are not affiliated with a particular university that successfully apply and compete for and win NIH funding. So I think it is the case, however, that to be competitive for NIH funding, you need a set of, a strong environment in which to do the research. And you need relevant expertise and capabilities. And so we foresee building that over time, in particular once we hire those PhD level scientists. At the outset, the idea is to co-apply with academic investigators. So we wouldn't necessarily be applying on our own, but we'd be partnering with a researcher at Hopkins or Mass General or University of Michigan. Eventually, though, we do believe that we can build up the capabilities of CF Labs to be competitive. And in fact, there is oftentimes in NIH grants a component of the score that has to do with innovation. And certainly, I think there's a strong argument to be made that doing research in CF Labs is definitely highly innovative. Dan, anything else you want to mention about that? Okay. All right. Chordoma tissue is rare as moon dust. Does the foundation have sufficient tumor samples of all varieties or more samples needed. I think I'll just answer that quickly. I think the short answer is there's always a need for more tumor samples, in particular for rare subtypes. And so if resources were unlimited, we would be focusing a lot more on trying to acquire those samples. The reality is that it takes an ongoing commitment of resources and ongoing infrastructure to be able to effectively collect tumor samples. And at the moment, that's not where we're prioritizing allocation of resources, not because it's not important, but because there are things that are of greater need, like CF labs, et cetera. Additionally, we have already acquired a pretty substantial collection of samples. And so the focus now is on making the most of the samples that we already have, providing them to the multi-omics projects that I mentioned earlier, et cetera, and as well as other projects.
Yeah, exactly. I think, the multi-omics projects, once we understand perhaps what the molecular subtypes of chordoma are, that might reveal potential gaps in our biobank that need to be filled. So just wanted to mention that. Yep.
Have mRNA vaccines or mRNA advances opened up possibilities for the approach, for this approach to be used with chordoma? So I think within mRNA advances, there's, I think it's worth differentiating. There are a set of class of therapies that are being developed that actually directly target RNA. And so we're actually exploring applying those to Brachiary to be able to block or eliminate Brachiary by targeting it at the mRNA level. The other area in which advances have been made with respect to mRNA is I think, which has been very much kind of in the fore or in the zeitgeist recently, is mRNA vaccines, a la Moderna or BioNTech, and those require the identification of a tumor antigen, so something that the immune system can recognize on the outside of chordoma cells. And in fact, that's a very, very good question, because we'd like to be able to leverage mRNA vaccines or other therapeutic vaccines for chordoma, but we have to first identify chordoma-specific cell surface proteins. That's not something that is initially going to be a focus of CF Labs. There's a project ongoing that we've gotten started and are actually now raising funds for at University of Toronto, where they are attempting to identify chordoma-specific cell surface proteins. A role that the lab could play is perhaps in validating those surface proteins in chordoma. Dan, anything else on mRNA vaccines? Okay, favorable response in terms of proposals being reviewed. So in the scale or in the launch phase, we have been focused on kind of standing up the lab, tackling an initial set of projects that have kind of been in the backlog. We've taken a passive approach and not actively solicited proposals because we haven't had the capacity to pursue them. However, even absent specific RFP, the list of concepts is already queuing up. And so the plan is, as soon as capacity allows, to put out an RFP. And we've seen with our drug screening program, when it was built, it drew out of the woodwork lots and lots of concepts that we didn't even know were out there. So we expect the same will be true when we start proactive outreach to the research community. But the key is we have to have the capacity in place to be able to act on the concepts that come in. All right, final couple questions. Talk a bit more about using sequencing and genomic analysis to predict which chordoma subtypes are likely to respond. Dan, you want to address that in any way?
Sure. Yeah. I mean, in a nutshell, what we're going to do is experimentally understand which chordoma models respond or don't respond to a particular treatment, such as afatinib, for example. And then we have all of this kind of model profiling, model sequencing data that we'll have generated that tells us what the genomic structure is of each model, what genes are turned on and turned off in each model. I know I know the person who's asking this question knows this, but I'm speaking kind of in more scientifically terms for the rest of the folks here. And what we want to do is kind of understand, once we have all of that data separately, can we take that data for looking at the various mutations in chordoma, the various genes that are turned on and turned off, and understand whether there are any differences in that can explain whether a model responds or doesn't respond. So can we kind of develop predictive signatures in that data that associate with response or non-response experimentally? And then if we can do that, can we then validate that model on another data set? So that can be existing mouse model data that we have with afatinib. That can be patient samples that are collected on a clinical trial where both patient sequencing data and patient response data exists. And so if we can develop that predictive signature and then validate it, I think it could be incredibly useful for chordoma patients. So that's kind of the idea.
Perfect. Okay, final question. What are the plans with respect to combination therapies?
Yes, great question. And the answer is yes. We are currently testing combination therapies, and we plan to do a whole lot more of it. I think that's ultimately where the cancer field, research and treatment field as a whole is already heading. And we are hopping on that train and trying to enhance the magnitude of responses and extend the duration of responses through rational combination therapy approaches, such as what you mentioned there with an immunotherapy and a chemotherapy, for example. So great question, and resounding yes.
Absolutely. I think just to underscore, the key is rational. You can imagine there being limitless number of potential combinations, and I think a challenge is that because animal experiments are so time intensive and costly, there's only so many that can be done. And so we really need to make sure that we've got a very strong, rational, robust, well worked out concept based upon work in cells to be able to design an animal experiment that's likely to succeed and that's likely to be a good use of funds. So I think that's hopefully a concept that's been evident throughout the presentation, but maybe just one to underscore and end on, which is that not only is CF Labs enabling us individual projects to be faster and more efficient, at a macro level, it's going to allow us to be smarter and more focused in how we deploy resources so that we can only advance the most promising concepts and the most well-justified and rigorous concepts into mouse testing. And then once we've got the results of testing drugs in mice, do further experiments to really make sure that that a concept that goes into the clinic is truly well justified, is very well worked out, both in terms of the drug or combination, dosing, et cetera. Because we're really only going to get so many opportunities to test drugs in the chordoma patient population. We've got to make sure that we're the shots on goal we're taking are good ones. And moreover, I think we all feel a strong ethical obligation to the community that we serve, to serve up and make available, really good concepts that patients are likely to benefit from. So that concludes our questions. We so appreciate the really great questions that came in. We appreciate everyone's interest and attention. This is something that is very near and dear to our hearts. We're extremely excited about. As I said a couple of times, that what we see is so powerful about CF Labs is, for the first time, being limited no longer by the structure and the constraints of the existing biomedical research ecosystem, academia and industry, but but being limited solely by biology and by the resources and will to make the research go. And we're so grateful to everyone who is making that possible. And we are excited to partner with many more of you going forward to continue to drive this forward and even to accelerate the work and allow the lab to live up to its full potential. So thanks so much for joining. Please, if there are any additional questions, comments, feedback, if anyone would like to get involved in pushing this work forward, please, we welcome you to be in touch. Take care.